Assalamu alaikum students. Today we will discuss life cycle of bacteriophage, a topic from 11th class biology, chapter number 5, variety of life. This is very important topic. In this topic we will discuss about life cycle of bacteriophage. There are two type of life cycle, lytic life cycle and lysogenic life cycle. Lytic life cycle is called master slave relationship and lysogenic is host guest relationship. Lytic life cycle is master slave relationship because in this life cycle bacteriophage cause destruction in bacterial cell. The lytic cycle result in the destruction of the infected cell and its membrane. Bacteriophage that only use the lytic cycle are called virulent phages. That kind of bacteriophage that are only doing the lysis or lytic cycle are called virulent phages. Lysogenic. In the lysogenic cycle, this is also called host-guest relationship because in this life cycle, to a certain limit, bacteriophage live peacefully within the body of its host. Phage DNA is incorporated into the host genome and it doesn't harm its host. Lytic life cycle. First, we will discuss lytic life cycle. It consists of four stages or four steps. The first step in this life cycle is attachment. Bacteriophage, first of all, find a host and attachment place on it. Penetration, then it penetrated. Third, multiplication. And fourth, lysis. First step, attachment. Bacteriophage replicate only inside the bacterial cell. That's why it is called bacteria eater. The first step in the replication of a bacteriophage is its attachment. Attachment is also called adsorption to host cell at receptor site. It means bacteria attach only a specific point. Bacteriophage is attached only a specific point of its host surface and that specific point is called receptor site on the wall of the bacterium. As you can see here, during attachment, weak chemical union between virion and receptor site take place. Next step is penetration. In the next step, penetration, the tail release the enzyme. Here, tail of bacteriophage release an enzyme and that enzyme is called lysozyme. This is also a short question. The tail of bacteriophage release an enzyme. That enzyme is called lysozyme to dissolve a portion of the bacterial cell wall and that enzyme, that is lysozyme, dissolve the portion of cell wall. Here you can see a marked area. First step, bacteriophage is coming for attachment and then it will do the next step, that is penetration. Landing of bacteriophage, attachment. Now the tail sheet contract. The tail is made up of two parts, that is sheet and core. The tail sheet contract and tail core is forced into the cell through cell wall and cell membrane. Sheet contract and the tail core is forced into the cell that is bacterial cell and it pass through the cell wall and then cross the cell membrane. The virus injects its DNA into the cell just as the syringe is used to inject the vaccine. The protein coat which form the phage head and tail structure of virus remain outside the cell. Here you can see again landing, attachment, tail contract and DNA inject inside the cell, but the head and tail mostly remain outside the bacterial cell or host cell. Many animal viruses, however, enter the host cell as a whole. Here you can see again two steps has been completed, that is attachment and penetration. The marked area is showing the next step, that is multiplication. In multiplication, basically replication occur or the bacteriophage take the control of biosynthetic machinery of its host and order it to manufacture head and tail for bacteriophage instead of making their own part. Immediately after entering the host cell, the viral nucleic acid takes the control of the host biosynthetic machinery and induces the host cell to synthesize necessary viral component. And what are these necessary viral component? That is DNA and protein, and start multiplying, as you can see here. Now lysis about 25 minutes after initial infection, approximately 200 new bacteriophages are formed. Bacterial cell burst due to so many number of bacteriophage, bacterial cell burst. That is, it undergo lysis. That's why this is called lytic cycle, because it causes lysis of its host, breakdown of its host. Here you can see again the whole cycle: attachment, penetration, multiplication. In multiplication, synthesis, duplication of DNA, replication of DNA take place, head and tail portion are formed, and then assembly of head and tail portion take place, and at the end lysis of host cell and release of bacteriophage take place. Newly formed phage are released to infect the bacteria. Another cycle, the lytic cycle, began. Again and again, they can cause the lytic cycle. The phage which causes lysis of the host cell is known as lytic or virulent phage. What is virulent phage or lytic phage? That bacteriophage or that phage which causes lysis of the cell, breakdown of host cell, is called virulent phage. The next is lysogenic cycle. When we will discuss lysogenic cycle, we will repeat the same step, that is attachment, penetration, and but there is a change in multiplication. There is no lysis take place. They live peacefully. Here you can see lysogeny, that is lysogenic cycle. All infections of bacterial cell by phage don't result in lysis. In some cases, viral DNA, instead of taking over the control of host machinery, become incorporated into the bacterial chromosome. It means this is the same cycle in which the first two steps are same. Attachment, we can repeat that step here again. Attachment, penetration. But instead of multiplication, we can use this content. In some cases, viral DNA, instead of taking over the control, there is not master slave relation, there is host guest relation, become incorporated into the bacterial chromosome. Phage in this state is called prophage. That phage which cause lysis is called virulent phage, and that phage with that doesn't cause lysis is called prophage, and this process is known as lysogeny. And this process is lysogeny. Here you can see lysogenic cycle: attachment, penetration, then live peacefully with host and it will multiply with host. But sometime in this cycle, while living peacefully, due to some environmental factors or due to some mutation, suddenly the lysogenic cycle convert into lytic cycle, then that stage and cause lysis, and that stage is called induction. With that, we have completed our topic. Thank you for watching.